Good morning, good evening, good afternoon, or good night, depending on when you're watching this video. This is Mr. Messina, ready to present another biology lesson. This one in particular is about the individual protein structures that actually help things cross through a cell's membrane. So without further ado, let's get rolling. Now, in the previous videos, we've talked about different types of solutions, and the one immediately prior to this, we talked about the cell membrane itself, which you can see right here. Just as a quick review, of course, the cell membrane is made of phospholipids, which have a phosphate head, which is right here, which is hydrophilic, which means it's water-loving, and then it has fatty acid tails, which are right here, which are hydrophobic, which of course means water-hating or water-fearing. So you can have water up here, let's see, and then you can have water in here. And this might be the cytosol or the cytoplasm, and this is just the outside world. And that's just the outside world. This is just kind of breaking them apart. So the membrane itself, in addition to being made of this phospholipid bilayer, because you have one layer here and then another layer here, you'll notice there's a bunch of purple and some red things over here that are proteins that actually help things move in and out of cells. So a lot of students are curious, what kind of things actually move through cells? The first thing are hydrophobic molecules. These are hydrophobic, meaning water-hating or water-fearing. And these things like oxygen, CO2, nitrogen, they'll pass right through the membrane without any issues whatsoever. You also have small uncharged polar molecules like water, glycerol, things like that. Those will also move right through and without any problems. But large uncharged particles as well as polar molecules, that's going to start having trouble. And some of the ions, some of those don't really pass through very well because of their charge. So they need a little bit of help to cross the actual membrane. Now, we talked about it before where to cross a membrane you typically need some kind of high concentration and you're moving to a low concentration. Now, even though that's the case, there are still these kind of ions that are not really getting through the membrane very easily. So they diffuse through a series of protein channels. And those channels are specific to whatever ion they are, and they kind of look like this. It's quite literally like a hole in the membrane that allows these specific ions to pass their way through. This is the same for larger charged particles as well, so not just ions can pass through them, but an ion channel is for only ions. For these large charged particles, they have their own set of channels that they have to find in order to pass through. Now, a hallmark about facilitated diffusion is it takes no energy. None whatsoever. It's a form of passive transport. And we'll talk a little bit about the difference between passive, which is like facilitated diffusion, and then active transport, which you'll see requires an energy expenditure. So, let me get this cleared up a little bit. As you can see right now, facilitated means with help. So facilitated diffusion means it just takes a little assistance on the cell's part to get these things through it, but not a whole lot. They're still moving from a high concentration to a low concentration, as you can see right here. Now, active transport requires a little bit more ingenuity. You see, sometimes cells need to move molecules against the concentration gradient. That means you may have a high concentration of something in here, so that's a high con. And then you have a low concentration of it, which is low con. But you still need more of whatever we're talking about in here. You may still need some kind of a higher amount in this area over here. So active transport is designed to move substances that are of low concentration through the membrane against the concentration gradient. So what happens is when one of these little molecules bind to the protein changes shape. So you can see the difference between this shape and this shape right here. You can see how they look very different because they actually are a little bit different. In this particular case, you have a protein that opens up this way, the molecule comes in between my hands and then changes shape this way, depositing the molecule on the inside of the cytoplasm. So a lot of times these are referred to as protein pumps because they are proteins and they actually pump against this gradient here. However, these pumps cost energy in the form of ATP, which we'll learn later. We learned before that ATP is produced in the mitochondria, but we'll learn about it more in depth when we get to metabolism. So you can tell that this right here is a low concentration to a high concentration gradient. So this is against the gradient itself. And it requires ATP, as you can see by our little cartoon. So that structural change that I was talking about before, that shift from this shape to this shape or vice versa, that's actually called a conformational change. And it's just a fancy word that some biologists came up with to describe that physical form change that these proteins go through when they actually pump things inside. Now, like I said before, there's passive diffusion. There's also active transport. Those aren't just the only two ways to actually get in and out of the cell. Cells can actually do endo and exocytosis. Endo means in, exo means out. So endocytosis means the cell is actually bringing something into itself. So you can see this is the environment and this is the inside of the cell, so this is the cytoplasm. You can see over here, you can see the pathway of endocytosis. Quite literally, just kind of like a little pocket of food comes, collides with the membrane, and the membrane actually makes this little vesicle. And this vesicle just kind of floats around in the cytosol, still kind of separated from the rest of the cytosol because it has this little membrane bubble protecting it. So in order to get at this little food particle that's in this vesicle, well, some other vesicle has to come in contact with it. This right here is a lysosome. So it's its own little membrane-bound vesicle. Those vesicles will collide and eventually merge in order to be this shape right here. So when those two vesicles merge, that's when the lysosome can actually take action and do something about that food molecule we were just talking about. So that's endocytosis. Like I said before, endocytosis is just grabbing onto something from the outside world and bringing it in, giving it a little vesicle. Exocytosis is when you're moving it out. So exo is out. You can see it right here, this kind of group, these two vesicles form together, that food was digested, and then it has to go out. So all those little particles are still trapped in the vesicle. The vesicle then merges with the membrane and then expels all those little things right out of the cell in that general direction. The Golgi apparatus, which we talked about before, is a master of producing these kind of vesicles. Not necessarily full of digestion products or anything like that, but the Golgi produces these vesicles, and these vesicles always go to the cell membrane in order to be let out of the cell itself. So like I said, getting through the membrane, you have passive transport. And that's simple diffusion, which is where things are just moving through the membrane. And that's nonpolar hydrophobic molecules. And that's like oxygen or CO2. Those things, they don't require energy. They don't need anything in order to get through it and make their way through. Lipids are another good example. Lipids are those oils that we talked about that the membrane is actually made of. They run from high concentration to low concentration. So they're going from high concentration to a low concentration. And then you have facilitated transport, which is the same kind of ball of wax where you have high to low concentration shift. But these are polar hydrophilic molecules. That means water-loving, and they can't interact with the membrane itself. So they need a little bit of help to get across the membrane itself. This can be done through a protein channel. It's, like I said before, high to low concentrations. And that brings us to active transport. Active transport is diffusion against the concentration gradient. That means we're going from low to high. I'll say that one more time. We're going from low to high. And that requires some form of protein pump of some kind, as well as it totally requires ATP. Definitely requires this stuff. It requires some form of energy. So you have passive transport and active transport. This one requires no ATP, and this one, of course, does require ATP. Think active ATP. This is a nice little summary cartoon that we have, so we can show you all the different types. So simple diffusion is just moving right through that membrane, like I said before. Facilitated, you need a protein or something to help it along, but it still doesn't take any energy. Meanwhile, down here, you have active transport, where you totally need some kind of energy because we're going from low concentration to high concentration. Whereas up here, we're going from high concentration to low concentration. Well, that's all I have for you guys today, or girls. Tune in next time as we continue our parade of lectures through biology. This is Mr. Messina signing off. Have a pleasant day, evening, or whatever, whenever you're watching this.